Fertilization is the specific biological process where a male sperm and a female secondary oocyte combine to form a single-cell zygote. This union restores the normal diploid number of 46 chromosomes, merging genetic material from both parents to initiate the development of a unique individual. Success depends on a highly regulated logistical sequence, demanding that specific transport mechanisms and biochemical triggers align within a narrow window of time. The sequence begins at mid-cycle, prompted by a sudden surge in luteinizing hormone, or LH, from the pituitary gland. This hormonal peak causes a mature vesicular follicle on the surface of the ovary to rupture, expelling its contents into the pelvic cavity. The released cell is a secondary oocyte, which enters the reproductive tract suspended in metaphase 2 of its second meiotic division. This animation of the rupturing follicle shows that the oocyte is not a bare cell. It is heavily shielded by an inner glycoprotein coat called the zona pellucida and an outer layer of granulosa cells known as the corona radiata. To reach the oocyte membrane, the male gamete must possess the specific enzymatic tools required to breach both of these defensive perimeters. This macroscopic footage shows the fimbriae, finger-like processes of the uterine tube, sweeping across the ovary to capture the oocyte and pull it into the infundibulum. Once inside, fluid currents generated by the cilia of the mucosal cells sweep the oocyte deeper into the tube. Rhythmic muscular contractions of the tube wall, known as peristalsis, then propel the oocyte toward the uterus. The target is the ampulla. Highlighted here, the ampulla is the widest part of the uterine tube and the primary site for human fertilization. With the oocyte now positioned at this spatial destination, the narrative focus shifts to the journey and maturation of the male gametes. At ejaculation, approximately 200 to 300 million sperm are deposited in the female reproductive tract. As the numerical counter shows, the attrition rate is staggering. Millions of sperm are immediately neutralized by the acidic vaginal environment or trapped in the mucosal folds of the cervix. Of the hundreds of millions that start the journey, only a few hundred surviving sperm ever reach the site of fertilization in the ampulla. This massive loss functions as a biological filter, ensuring that only the most robust and motile gametes arrive to encounter the oocyte. This schematic maps the surviving sperm's route as they navigate from the cervix, through the uterine cavity, and into the uterine tube. While sperm have their own flagellar motors, they do not swim this entire distance through their own power alone. Their passage from the cervix to the oviduct is primarily assisted by the muscular contractions of the uterus and the uterine tubes. This makes sperm migration a coordinated effort between the male gamete and the female reproductive tract. Even upon reaching the oocyte, freshly ejaculated sperm are not yet capable of completing fertilization. They must first undergo capacitation. As noted here, this is a 7-hour conditioning period involving biochemical interactions with the mucosal surface of the uterine tube. During this time, a glycoprotein coat and seminal plasma proteins are removed from the plasma membrane overlying the sperm's acrosome. Capacitation also triggers an influx of calcium that alters the mechanics of the flagellum. This creates hyperactivated motility, a violent whip-like thrashing seen here. This functional maturation is the final prerequisite. Only capacitated sperm have the physical and chemical state necessary to breach the oocyte's defenses. The timing is critical. This oocyte in the ampulla will remain viable for roughly 24 hours post-ovulation before it begins to degenerate. This brief lifespan must overlap with the arrival of sperm that have already completed their own 7-hour capacitation delay. When the gametes finally meet, the sperm are confronted by two distinct structural barriers, the corona radiata and the zona pellucida. The encounter is a race against a biological deadline, where the sperm must execute a specific series of biochemical reactions to reach the oocyte surface. The first perimeter is the corona radiata, a dense matrix of granulosa cells held together by hyaluronic acid. The sperm uses its hyperactivated motility to physically force its way between these cells. This process is aided by the release of the enzyme hyaluronidase from the sperm's acrosome, which disperses the follicular cells to clear a path. Defeating this first barrier requires a combination of sheer physical propulsion and targeted enzymatic dispersal. Once through the corona, the sperm reaches the zona pellucida, a thick extracellular glycoprotein coat. This scanning electron micrograph shows spermatozoa physically bound to the spherical surface of the zona pellucida. This molecular diagram maps the specific recognition event required for fertilization. The sperm must identify and bind to a precise glycoprotein receptor, known as ZP3. This receptor-level contact is a critical checkpoint, preventing cross-species fertilization and ensuring only a human sperm can initiate the next phase. Binding to ZP3 acts as the final trigger for the acrosome reaction. As detailed in this technical source, the sperm's plasma membrane fuses with its outer acrosomal membrane, creating multiple perforations in the head of the sperm. These apertures allow for the release of a payload of proteolytic enzymes, most importantly, acrosin and neuraminidase. These enzymes literally digest a localized tunnel through the glycoprotein network of the zona pellucida. The sperm passes through this dissolved path, placing its head in direct contact with the plasma membrane of the oocyte. The biochemical gauntlet has been cleared. The gametes have made physical contact, setting the stage for the fusion of two cells into one. The moment these membranes fuse, the oocyte will trigger an immediate cascade of reactions that permanently seals the egg against any further sperm entry. In our next module, we will explore those immediate consequences, from the cortical reaction to the final formation of the zygote.